Okay, let's start. So I am really excited to be here today to introduce two people actually who are in my lab, one of my groups at JPL. So Dr. Jessica Weber. So I met Dr. Weber actually like almost 4 years ago now. So we met right before the pandemic. So she works in the group with some other scientists in studying the origins and habitability of life here on Earth, which is a really important thing to study because as we can study it on Earth, we can begin to understand how it might occur and be happening elsewhere in the solar system or beyond. So we can study our own case that we know very well here on Earth to understand it elsewhere. So Dr. Weber comes from a chemistry background. She did her undergraduate at the University of Chicago and her PhD at MIT. And she's been here ever since. So she's now a research scientist at JPL. So a really great member of our group. And then Dr. Teese, I have not known her for as long, but she's been an amazing addition to our group. She joined last year, came from a geology background. She did her PhD in Australia at the University of New South Wales in Sydney. She has an expertise in organic geochemistry and astrobiology and in science education. And she really helped me with this program and gave me some tips and pointers because she had experience in science education, whereas I was pretty new to the whole world. So I'm going to let the two of them introduce themselves and kind of discuss their path, and then we will open it to questions with all of you. So Dr. Weber?
All right. I don't know what time zone all of you are in, but here it's morning. So I'll say good morning, everyone. As Tess said, my name is Jessica Weber. I'm initially from the Midwest in the United States, from Flint, Michigan. And I'd always really liked science and thought that was very exciting. So when I went to college, I pretty quickly honed in on chemistry as my major. And in college, I had a ton of really excellent mentors who encouraged me to work in the labs at the university, in chemistry groups. And so I was able to get a lot of hands-on experience early in my undergraduate, working in labs. This was all very synthetic chemistry, like kind of like synthesizing drugs, sort of research. And at that point, I really decided that I wanted control over what I research, and I wanted to have a lot of input in the projects I research. And so in order to do that, I thought I really needed a PhD. And so I applied to the chemistry PhD programs, and my PhD actually is not in astrobiology or really anything related to planetary science. It's in this very fundamental, drug synthesis, designing, metal catalysts to make reactions faster, that kind of work. But a good part of my PhD was funded by a government agency in the United States, and that was highly collaborative, huge projects. I got to work with engineers and biologists and a lot of really cool people, and I really liked that experience. And so when I was looking for postdocs, I was looking for a really collaborative field where you could work with different people, work on big, hard problems. And what's harder and cooler than trying to understand the origin of life? And that really led me to astrobiology. And so I did my postdoc with Dr. Lori Barge, who's one of the PIs in the OHL at JPL. And I liked it so much, I decided I wanted to stay. And so I applied for research science positions and was very lucky to have my role now. And I would say my role now is about half-time research. And, this is chemistry, origin of life, experimental in lab, on my feet research. And then I'm half-time working on, different, strategic NASA concepts, mission concepts, and stuff of the like. And I'm really excited to take questions.
Thank you. And Dr. Teese?
Hi, everyone. So I'm Bonnie, and I also had like a sideways path into astrobiology. So I actually never thought about doing science. I always thought science was for rich old men. And so it just wasn't a career path that I ever thought about. So I did not have the opportunity to finish high school, and I had to leave high school and work full time. And I did my equivalent of school in something called TAFE, which is sort of like an Australian community college. And then I got rejected from every university I applied to And so I had to do a bridging course for disadvantaged students where they help us gain the skills that we need to be able to study at university, especially if no one in your family's ever been to university or finished school, or if there are other issues that may prevent you from succeeding at university. So I did that course and then I started uni and I was actually studying English literature. And I was really fascinated with like science fiction and fantasy books. And I really wanted to do a PhD in science fiction. But the university that I was at required that you do a subject outside of your faculty. And there was this subject in the science section, and it was like called Life, the Universe, and Everything. And I was like, I recognize that book title. I'm going to do this subject. And so I started doing this subject, and the convener one day handed me a scientific paper. And that scientific paper was by an Australian woman named Abigail Allwood. And she became the first Australian and the first woman to lead an experiment on Mars. And I was like, oh my gosh, I didn't know that we could do that. So I added a science degree onto my arts degree. And then I started desperately trying to learn science, like very desperately. And so I did a master's degree before my PhD because I was still trying to catch up on all the science I didn't know. And I started really getting interested in the early Earth and how we look for life that's really, it's been so altered, it's not even recognizable as life anymore, and how that helps us look for life in places like Mars and other places across the solar system. So when I finished my PhD, I had no idea what I was going to do because in Australia, we don't really have an astrobiology program. We have a very young space agency and we don't have opportunities to get involved in missions. And so I saw a job advertised at JPL and I thought, oh, that looks really nice. I'd like to do that. And then so I applied and now I'm here and I'm studying organic compounds in hydrothermal environments and how they're relevant for life detection across the universe.
Very cool. I love that your two stories are so different and you both ended up in the same place down the hall from each other. So could you describe like what your average day looks like, just like a day in lab? Maybe Jessica, do you want to start?
Yeah, I can start with that. So I will say what I love about my work is that my day-to-day is really varied. So some days, so today, for example, I'm doing this with you all. I have to pick up some lab equipment at about 9:30. I have a 2-hour meeting. Like we're having like a very intense data workup. Students are sharing their raw data and we're going to dig through it and format it and make sure we understand yields and all that stuff. And I have another meeting with a student and then I've left the afternoon basically for me to be in lab. I have to grind a bunch of minerals today. So in order to use them for experiments tomorrow. So I've got to grind and sieve minerals. But I would say that this really isn't a normal day. So some days I'm in meetings all day discussing either experimental planning or mission planning. And then, for example, tomorrow I'm running experimental tests to test like material retention for a possible comet sample return mission. And I'm doing that all morning. We have like a system with a spring. You can load it up, you can put it under vacuum to test at least some kind of analog of space. And then, load whatever you want your target to be, what it hits, and then whatever you want your sample to be. And I'll probably do that all morning. And then I have a backlog of writing that I probably should do and editing. But it really does vary. When I was a postdoc, I thought my life was a little more consistent. I tried to be in lab in the mornings for a good couple hours. It was nice because if you were in early enough, you didn't have to fight for space on the glove box, which I'm sure Bonnie will also mention. And then I would try to schedule all of my meetings in the afternoon, but that's since I'm working on very different projects and I have collaborators in Asia and in Europe. So I've got to be very flexible of when I meet. So, yesterday we had a meeting with someone who's based in Paris. And I have regular meetings with people in Tokyo. And so that means that, you have to accommodate these people's time zones. California is not the only place in the world. So that means sometimes I have very early morning meetings or very late in the afternoon meetings, depending on the day. But yeah, like I said, the really beautiful part about this job is that it really varies day to day, and you can be very flexible in kind of how you structure your day. A lot of that is very much up to me.
Thank you. Bonnie?
Yeah, like Jessica, my days are extremely varied. And like Jessica, I also work with people in Europe and people in Australia. So some mornings my day might start with a meeting at 6 a.m. And then other days it might finish with a meeting at 8 p.m. And some days that really aren't great, it starts with a meeting at 6 p.m. and then ends with a meeting at 8 p.m., if I don't schedule well. But my days are also incredibly varied. So if I just think about the last 3 Mondays, this Monday, I was in the lab the entire day, setting up experiments, working in an anoxic glove box that we have that simulates the early Earth before it became oxygenated. But if I think about the Monday before that, I was actually in a mission design school, learning how to design missions, working with a team that we'd been designing a mission for 3 months. And we were in like an intensive week, like trying to finalize our mission. And then the Monday before that, I was on a ship in the middle of the Pacific Ocean, looking at deep ocean hydrothermal vents with a remotely operated vehicle that was down 1.5 kilometers on the sea floor, exploring hydrothermal vents. And I have to say, like, that variability and that opportunity was what had originally it excited me about science. I'm a field geologist, so a lot of the time, like, I might be in the lab a lot when I've got my samples, but I also get to go to the field a lot and experience lots of different places in the world that I might have never been able to see. So it can really vary depending on the time of the year. In Australia, field work was always in winter because you don't want to go to the desert in the middle of summer. But here in the US, a lot of the field work seems to be more in summer than winter. So summers can be quite busy.
Thank you. So another question, how do you think, so like your work are very varied. So like Bonnie, you're doing field work and lab work, and Jessica, you're doing mission concepts. How do you think the different aspects of your work inform each other? Like, how are you using lab work to basically inform the other parts of your job?
Yeah, I do think it's one big circle at the end of the day. With our, so I should say my lab projects are broadly focused on understanding where organics are in the solar system, which is very fun because that means I can work on any planetary body. There's probably organic material everywhere. And so I have a lot of variability in my projects, even worldwide, which is nice. And so with this, I want to constrain it and understand these analog systems experimentally in lab. However, in order to make that actually useful, it has to connect to these mission concepts and to what we want to see. So for example, if I'm working on a project related to Mars and understanding how organics could transport on Mars, that's really important to maybe integrate similar techniques that the rovers use to analyze these samples, or at least be aware of what the data from the spaceflight missions have to maybe constrain what I'm testing. And so not only do the space missions inform the work, but then the work can come back and inform, hey, maybe we need to look at this closer, or maybe we need to, send something else to further kind of constrain this variable. And I think that that's a really important part of, the research, is to see those connections.
Thanks. Bonnie?
Yeah, I would definitely echo what Jessica's saying. So for instance, the field site that I work on in Australia is 3.5 billion years old. And this site holds the oldest generally accepted fossils on the planet. And this is about the time that Mars would have been wet and potentially habitable. And so our field site is a site that people come to all across the world to learn how to look for life on Mars. So my PhD advisor, he would often host scientists from the Perseverance team from Mars Sample Return and bring them out into our field sites so that they could get a feel for how do you actually look for life in these really ancient environments where life isn't everywhere yet and where they've been so altered. So there's a fundamental research aspect that underpins all the practical space missions. And then we can't learn everything from the field alone because a lot of the instruments we use, you can't just do in the field. There has to be a lot of preparation. So we bring the samples back to the lab, and that's where we can really see things at different scales that then really give us a lot of knowledge about the processes going on in the universe, help us narrow down where you might want to look for life or signs of life, and help us think more broadly about where life could exist, where it could get started, where it might evolve, what those limits might be. One thing I've particularly enjoyed since moving to the US is the more translational aspect of the research. So in Australia, we did a lot more fundamental research and we were exploring a lot of questions because we were interested in them, which is very fun. But here it's very interesting to start doing more research that is related to space exploration and to the practical aspects of how you do astrobiology.
Thanks. So we have a lot of questions in the Discord. So one question is about, it's from Anna in Romania, is asking how you apply drug and pharmacy information to astrobio. And I guess like that's definitely a question for Jessica, but like also just more generally, how do you apply like your very non-astrobiological beginnings in education to your current work?
All right, so my take is that in your PhD, you are learning to run the scientific method. So there are so many transferable skills that you get in your PhD. Certainly, I can tell you a whole lot about nickel catalysis, which I don't think anyone wants to hear in the next 40 minutes, which is totally fine. But in my PhD, I learned how to run a project. I learned how to evaluate data. I learned how to write that data up and present it either in a paper or in a presentation at a conference. And I think that those are all really critical skills to science, and it's true across the board that you need it. And secondly, since I'm focused on constraining organics in the universe, that makes it really, really similar to the organic chemistry I did in my PhD. And both were experimental, so a lot of the techniques I use are still the same. For example, the main analysis technique we use is something called nuclear magnetic resonance. It is a huge workhorse in organic chemistry. You very rarely ever see a like scientific organic chemistry paper without NMR. It is like a more of a rarity than not. And so that technique, I had ran thousands of samples of that in my PhD, and that is something that I could bring to JPL and still use here. And so a lot of the actual lab techniques ported over quite well to my postdoc. I was just asking different questions. And so what I think about my work today is it is organic chemistry, but with a specific subset of conditions. In organic, like drug synthesis or catalysis, you want the reaction to work with high yield and high selectivity. And that is, you can pick any solvent, you can pick any reagents. For this astrobiology origin of life work, you have a subset of reagents that have been picked for you. It is what is reasonable on the early Earth. And so it's just narrowing down, the conditions that I can use, which I think is really cool.
Thank you. Jessica actually taught me how to do NMR, so she's a great teacher in that. So, Bonnie?
Yeah, I think a lot about the community that I grew up in regards to how education plays into astrobiology. And in the community I grew up in, education either wasn't valued or it wasn't accessible. And I wanted to make sure that my opportunity to get education was something that could play back into my community and help support my community. And one of the great equalizers or the great things that interest people in science, even if they have no other interest in science, is space research. The questions of, are we alone in the universe? Where do we come from? This is something that excites curiosity in everyone. And so I use a lot of the education that I've been able to gain to try to inspire other people in my community and try to help them become interested in things that are outside of their immediate world. Sometimes it works, sometimes it doesn't, but at least we're trying, I think. And I think it's with space in particular, because it is primarily funded by government organizations across the world, we really do have a responsibility to make sure that we're communicating things and that we're educating people and that we're using the research we develop to help lift communities up. So I really see it in tandem. The research that I do is something that belongs to the whole world, and so it needs to be used to educate people.
Thank you. Bonnie really helped me shape this program. So I think that Bonnie's ideals are kind of shining through in this program because we had a lot of conversations about it a few months ago. So another question, this is more of a research question from Volley, and that's how is the mineralogy of a planet and its potential for life habitability related?
Okay, so as far as we know it, for life, broadly speaking, you need a solvent on Earth that's liquid water, organic material, and then some kind of energy, broad strokes. And for those organics, we are all made up of a lot of complex organic molecules, to really, really deeply simplify things. And in order to get all of those complex organic molecules, you have to make them somehow through some kind of chemical synthesis. And so for my work, the mineralogy is especially important because we know a lot of these minerals are reactive with organic material. And this is, again, very focused on my research. There are a variety of geochemists and other people who are very interested in mineralogy. And I think Bonnie's even got a different take than I on this. But for me, I care a lot about what minerals are available to react with the organic material, so we can either get closer to an origin of life event or just really understand what kind of chemistry is possible. And by understanding the possible chemistry, then we constrain habitability, or like my work can help constrain habitability and help constrain, how close we could get to some kind of origin of life like chemistry.
Yeah, for me, I think about it in a slightly different way. I think about how the mineralogy can guide us to finding signs of life. So one of the things that I think about is how plate tectonics are necessary for cycling nutrients to the surface of a planet or a moon, or some sort of cycling transport event on Earth that's plate tectonics. And so I look for minerals associated with like hydrothermal areas you might see where there's going to be a lot of energy and nutrients concentrated. I would also look for minerals that are capable of preserving signs of life. So some minerals are really, really good at that, some are really bad at that. And so we can use the type of mineralogy that's present to look for that. We can also think about how a mineral might be altered. So olivine, for example, is a mineral that comes from the mantle, but when it's in a hydrothermal environment, it can be altered by water and become a mineral called serpentine. The alteration of olivine to serpentine produces a lot of energy that can then be used for life. And so we know if that alteration is taking place, that's an area that we should zoom in on to have a look at what's going on there.
Thank you. So Lena is asking, because Bonnie, you had said that science is something for old men, kind of was one of your earlier thoughts. Do you sometimes now catch yourself questioning yourself in a way and like wondering like maybe a man could do this better, or have you been able to come more to terms with things in your professional journey?
Yeah, so I'm very lucky now. The lab that I'm in is majority women, and that's a very different environment to some that I've been in. Coming from geology in particular, geology is often considered as like a man's science. So for example, in Australia, there are 250 male earth science professors and 20 women earth science professors. And so you are often the only woman in the room. And especially when you're young and you're early in your career, that often leads you to being discounted. It's even worse in the mining industry because the mining industry has a very large ratio of men to women as well, and that's in an environment where you travel away and you go stay there. And so that did lead me to question myself for a long time, especially because of my background. I did wonder if I belonged in science, and I do still wonder that. But then I also think probably these old men don't wonder that, and so maybe I should just try to fake the confidence of those old men and see how that goes. I think that we are so often concerned with like, should we do this because of the virtue of like who we are or what we bring to the table, but we don't necessarily think, yeah, I should do this because of the virtue of who I am and what I bring to the table. So I've been trying over the past few years to focus on that the things that I think about or that I might be bringing are a different perspective than most people, and that perspective deserves to be heard in this field.
A great answer. Thank you. Jessica, do you have anything to comment on how it is to be a woman in science?
Yeah, I definitely have had many experiences where I'm the only woman on a project or the only woman in a room. And, even there's projects at JPL where, I'm as a woman very outnumbered. And I think that it is it is something that is changing, and I am very hopeful for the future. But I think, to echo what Bonnie said, is something that I think about a lot is you can't make changes if you aren't in the room. And so like just staying in the field and being here is enough sometimes.
Definitely. And these two are both really big inspirations. So our lab is like structured to like people mentor people. So I mean, all the students are definitely very inspired by both of them, which is a great setup for everyone. So Sarah from Mexico is asking in a similar vein, what is the job environment like in the lab?
Okay, so I should say, and I have not fact checked this, but I've been telling people this because that's what someone told me. So a JPL is apparently about the same size as Disneyland. Rough estimate. I'll leave someone else to fact check that approximately. But there is a lot of different lab space here. So I just want to be clear that I think the OHL is really unique because we run what I consider to be a semi-academic lab. So we host group meetings, we have students at different levels, undergrad, master's, and PhD. And so we have kind of a variety of an environment that is academic adjacent. And I think that the lab is, open and welcoming. But I would say at JPL, there's a variety of laboratory experiences you can have. There's more engineering labs where they're, working on developing instruments. There's more, let's call it field heavy labs, which I think is more closer to Bonnie's PhD, where there's a lot of field campaigns. There's a lot of, rocks from different places in these labs. And then there's labs that are focused on, say, like one setup. So it's like we've made a giant vacuum chamber to, use to simulate this one airless body. This is what we're going for. And so I think that our lab is really, unique. We have a lot of experimental setups. We also have a lot of analysis tools in lab. And we also have very, strong connections to local universities such as Caltech, CSULA, and Citrus College.
Yeah, I would also add one thing that I think is really cool and unique about our lab that I wish more labs would do is that there's a lot of non-hierarchical training. And so the people who are experts in something are respected as experts in something, and you'll have master's students training postdocs, and you'll have a lot of information exchange. And I think that that is really nice to value people for their expertise, regardless of what stage of career that they're at. And it really showcases the value system in the lab, which I think is very special. I also think that, as Jessica said, the lab has a lot of relationship to other universities and community colleges. And there is a huge emphasis in our lab on education and outreach, in addition to science and missions. And there's a huge emphasis on communication in the lab between lab members. And so we have quite, I mean, I think it's a large lab, but I come from a country with a very small population, so it's possibly not a large lab by American standards. But that requires a lot of communication between people and talking about instrument usage and making sure that we're sharing things and that we're communicating constantly. And so I think that that's a really good skill in our lab. It's very collaborative and communicative.
I just want to add two things. One, we have a really excellent undergrad student who works with us, and in the whole lab, we need to change the gloves on one of our glove boxes. And out of everyone, there's only 2 people in lab who know how to do that. And one of them is the student, Matt. And so we're waiting for Matt to get back to basically train the rest of the lab. And then 2 is a huge part of what I do in the lab is coordinating lab safety and running lab meetings. And I think that, finding, especially in lab where stuff can go wrong, there's a lot of chemicals and people and, JPL is a big space and it's not always our people who are in lab, it's like there's labs around us. And so communicating what's going on is really, really critical for good, safe lab work.
Thank you. It's a great answers. So Lena is asking a few questions. So what kind of detection methods are there to detect organics? Does it depend on the organic being detected? And is it difficult because you don't always know what to search for in the beginning, so it's hard to develop the technology?
Okay, so you're going to have to remind me about the questions because I'm going to forget at least one of those. So I would say that we are not doing technology development for analysis for instrument usage necessarily in our group. And you can do at least some back of the envelope kind of predictions of like, if I put in A, I should be able to get B, C, and D. Like F and G are not possible if I don't add this thing. And so it's like, you should be able to at least narrow down your product space. With these organic reactions, you're putting them in for a purpose. We're not just like dumping a bunch of stuff in a flask and like crossing our fingers. We're trying to make something specific. And so we do have some idea of what to expect. Of course, there's always surprises in organic chemistry. Weird stuff happens. You can't control the reaction. So to say, okay, so from the bottom, we do have some kind of prediction of what we're looking for. We're not always right, but we at least have something going in. And then I think, what are our tools for organic analysis? Our big workhorse is the NMR technique I was talking about earlier. We also have access to Raman and IR spectroscopy, which can help. I consider those like functional group handles. It doesn't give you kind of the picture of the whole molecule that NMR would, but you can get at least some information about what kind of organic material is there. And then we use UV vis spectroscopy as well. And so I would say those are 4 main techniques. If we ever want to do something special, we can find a lab friend or send out samples. And then Tess, what was the second question?
Does it do the methods vary depending on what organic you're trying to detect?
Yes. So I would say 90% of the time we at least try NMR. But NMR is a liquid solution. So that means that 2 things is that you have to get your product into solution and then it has to not decompose while it's in solution. And sometimes those are very big asks for organic material. And so for the stuff that's not soluble, we have to do some kind of solid state analysis. We have to try other solvents. There's, workarounds. And then for the stuff that's decomposing, we need to really do something in situ in our lab. So for one of my experiments, we just couldn't get the NMR to work. I won't belabor the point, bad things were happening. The NMR spectra was not helpful for us. So we ended up turning to UV vis because we needed something in lab that we could walk from the workup to and just run immediately. I think that answers all the questions. But
yes, thank you. Bonnie, do you have anything to add?
Yeah, so before I was in this laboratory, I had never used NMR, so I used a lot of other techniques to look for organics. So 2 of them Jessica mentioned, Raman and IR. but the main instrument that I used was something called gas chromatography mass spectrometry. And that's a very similar instrument to the SAM instrument on Curiosity rover that there's a lot of publications about it, finding organic compounds in the sheepbed mudstone. And so when we are looking for organics in natural samples, like geological samples, we have an idea of what kinds of compounds might be in there or how they might be detectable based on the age of the sample or how much alteration has happened. So there are some compounds that are very resistant, they like to hang around, they're hard to destroy. And so those compounds we know will survive in the older rocks, whereas there are other compounds that are less resistant and they don't like to stick around very much, and they'll just break up if you apply some UV or some heat to them. And so you can decide when you're first looking at a geological sample what kinds of things you might think would be present, and choose between some different kinds of instruments. And that knowledge about the context of the sample is another way that we think about the reliability of any organic compounds that we find within a sample, because a lot of the problem with organics is there's organics everywhere, and sometimes organics get into a rock that aren't the organics that were in the rock when it was deposited. And so you need to know a lot about the context of the rock or the sample to understand what's really from that rock when it was being laid down.
Thank you. So Malaya from the US is asking, I know it's highly variable, but on average, how many hours a week do you work?
So this is, let's just say travel notwithstanding, because let's not factor in my like 12 hours, so like a 12-hour flight into this. I would say the job that I have is a 40-hour work week. The reality with science is that I do end up, every so often I'll end up editing something, or there will be something, weird that comes up, especially when you're kind of maybe working more flexible hours. But I try, I really try. I do not work weekends. And then JPL has the work schedule is called like a 480, I think, or like a 940, something like that, where you have every other Friday off. So you work 9 hours Monday through Thursday, and then every other Friday you work an 8-hour day. And on my days off, I do not work. And I think that having work-life balance is really important. And I'm not saying that I do it perfectly every week, but I think that the common misconception of science is that, you are working these 800 days, you can't have a life. And I think that in our group, we encourage people to take vacation. And even if I maybe, work a little more than those 40 hours, I do take my weekends and I do take my days off.
Yeah, for me, I guess now that I'm a postdoc, I work closer to 40 hours a week. Although sometimes it is over, like Jessica said, especially when you're doing field work, because you've got to work every day on field work because it costs a lot of money to be out there. But when I was doing my PhD, I did have to work a lot more because a PhD salary or a stipend is very low. And this is something that there's a lot of protests about across a lot of different countries. And so sometimes it can be hard to afford things if you don't have money saved up before doing a PhD. So I will be honest that during my PhD, I worked my PhD full time, and I worked 2 to 3 jobs on top of it to finance going through a PhD, which is not what should be happening. Unfortunately, that's the reality of that situation in a lot of countries. I will say the university that I did my PhD in raised the PhD stipend by $7,000 a year after I finished. So it's a lot better for students that are going through their PhDs now. Since I've been at JPL as a postdoc, I haven't had to do that. I work 1 job, very nice. And I am closer to 40 hours. I am still finishing off some work from my PhD that was left over. So sometimes I might work a bit more than that, but that's by choice. That's not something that's forced onto me.
Thanks. So Krimesh from Sri Lanka is asking for tips for becoming an astrobiologist when you're living in a country where they don't really have a lot of infrastructure already. Hey, Bonnie.
Yes, it is sometimes hard, especially if you're in a country where there's a very small astrobiological community. I think there's about 10 astrobiologists in Australia, maybe slightly more. And that can make the community really small. It was particularly difficult during COVID where we couldn't leave the country or even our state. So it was very hard to make connections with anybody. Um, I just really focused on the science that I was doing and what I enjoyed and making connections with people across the world. And I also thought about the ways that my work wasn't simply astrobiological, it had a lot of other implications as well. So the work that I do is also very important for early Earth geology, for paleontology, for understanding how the Earth's climate has changed and evolved over time, for understanding how the Earth's tectonics have changed and evolved over time. And so there's often multiple ways to view and frame your own work. And so you can think about how that might be valued in your home country. And then you can also use the impact of that work to try to convince your home country they should invest a bit more in astrobiology. I also found a lot of people, so during COVID, I was part of an early career group that was founded for people who were like the only astrobiologist at their institution. And it was to help us make connections and make a network of people. So we met every week during COVID. We still meet now fortnightly because we all decided to write a paper together. But having that community and having other people from other countries that were going through the same thing was also really helpful because I could talk to them about what they were doing, what it was like for them, and think about those opportunities. We did also lobby the Australian Space Agency to think more about astrobiology, but unfortunately it's not a priority of the government, and so they can't do that. But speaking regularly to MPs and senators and people in the space agency is always a good idea as well to try to enact the change.
Very cool. Jessica, do you have anything to add about tips to get into astrobiology?
Yeah, I just want to say, I think and I think Bonnie touched on this, but I think a silver lining of the pandemic is that there are so many, virtual workshops and virtual events like this to connect with other people. And that is, I think that I imagine that this is not a convenient time for everyone. And not all of these kind of events are going to be convenient for everyone, but at least being on the lookout for different early career groups and different workshops that are available is a great opportunity.
Thanks. So Nidhi from India is asking how research is different depending on the conditions, and like for places where it's different from early Earth, how do you like change laboratory conditions to simulate different planetary bodies?
Okay, so with your lab setup, you have to make some agreements because you're like lab equipment is expensive. And you are only going to set it up once. So you're not going to go, oh my gosh, today I want to change the whole gas system that costs 10 grand. Let's just do it on a whim. Once you kind of commit to that, you're in. And so, of course, in any laboratory setup, we are not going to Mars or Enceladus or, the moon. We are making a lot of assumptions, and you've got to pick what the most important assumptions are. So in a lot of the work for the early Earth, an important thing is being oxygen free, which is why we have these 2 oxygen free glove boxes. And then we can adjust our water solutions to be at a pH we want to test relevant to the early Earth. We can pick minerals relevant to the early Earth, and we can pick salts and organics that are at least believable on the early Earth. And so for this, I would say a lot of our lab setups are more, let's call it general. Like we don't have like the Mars chamber, for example, where there's some labs that are dedicated to say, hey, we have the pressure gas mix and mineral composition of Mars. This is where you do your like closest to Mars experiments. Whereas our chambers these glove boxes are much more general use. And a lot of what we do, so let's say we're talking about like a water solution. So we could compare early Earth, let's say early Mars, and then ocean worlds like moons of Saturn and Jupiter, something like Enceladus and Europa, based on data that we have from spacecrafts, based on modeling. There's some believable conditions on these, worlds, say varying the pH from acidic to basic. There's different salts that are believed to be in the oceans. And so that's stuff we can very, very easily. So we could set up an experiment that's early Earth based on pH and mineral, something that's Enceladus based on pH and mineral. And actually, this is a lot of what Tess has done in her work as well. But yeah, I think that, our lab setups are a little designed to be a little more general, and we actually mostly use the experiments to be as specific as possible, but it's all an approximation.
Yeah, and usually, you would list the limitations of your study in your research paper as well. So, some people study very extremely cold bodies in the planet, in the solar system, which are, negative several hundred degrees Celsius. And so there's a limit to how cold we can get things. And so they will just note in the paper the limitations of that study and think logically about what that limitation might mean for the generalizability of their data. So as long as you're honest and think critically about what the limitations of what you are doing are and communicate that, I think that everybody understands. We can only do so much in the lab.
Definitely. So Seetham from Sri Lanka is asking about, while we're able to simulate the prebiotic earth in labs, how does the scaling down of the environment affect the chemistry and interactions that we try to simulate?
Okay, so just to reiterate Bonnie's point, we are always making assumptions. If we want to, and what we've chosen to do is run a large swath of experiments inside of one, kind of like smaller chamber. And again, this is shared lab space. We are sharing these glove boxes. It would be very, very rude of me to say, hello, everyone. I would like to run an experiment with a ton of sediment, taking up this whole thing. Like it's really important for us to get variability and understand how the different conditions affect our experiments. I will say we have varied the scale of our experiments, especially some of the amino acid synthesis, and we've seen that we still get similar results. And I find that what is maybe more important than scale is concentration and relative concentration. So less so of like, is this the whole ocean, and more so of, is the organic at a comparable concentration to our mineral? And are those at comparable concentrations to the ocean? And so that was a bit of a varied answer, but I hope that at least, gets at the question.
Bonnie, do you have anything to add?
No, I think Jessica covered it.
Gotcha. So Joshua from the Philippines asks that since we only really have one example to study the origin of life, can you talk about the aspect of your research that could account for the possible many ways that life can arise in other planets to make sure that we're not looking for an exact copy of the biology that we see on Earth?
Yes. So first off, I want to say that we should use our example of life on Earth. We can't go too sci-fi with it. There's of course a lot of possibilities, but we do have this one very successful example that we should study well. So I think that that's an important thing to kind of stay on track. And I think that an important thing is that in astrobiology, there are people studying widely different theories for origin of life and widely different aspects of origin of life. So there's people who are studying, like, say, us, who are studying organic synthesis and preservation. How can we make it? How can we keep it? There are people that are studying how to encapsulate those organics into some kind of cell-like structure. There are people who are looking at making like large polymers, like protein-like molecules. And all of these come together to tell a story. And I think that that's why it is so important to have a variety of researchers from different perspectives studying these problems, because everyone is approaching it differently. And so I think that there's also a ton of literature I won't belabor the point. I will let Bonnie talk, I promise. There's a ton of like literature and discussion on biosignatures. And while some of those are very earth-based, there's also people looking at something called agnostic biosignatures. And these are believed to be overall like just needed for life. And some of that is looking for molecules that can function like DNA functions for us, looking at like lengths of membranes. This is out of my research, but you know, there are people looking at these agnostic kind of concepts. And so that's, you know, I think that's a broad picture of, you know, of what we're looking at. You know, we have to start from somewhere, and life on Earth is a very reasonable starting point.
Yeah, I would say I'm not an origin of life person. I more look at what happens to signs of life if we try to break them down. But I think thinking about where life might have emerged is important for trying to find those signs of life. And so we don't know how life emerged on Earth. And so there's, you know, as Jessica said, there's lots of research into different ways that it could have possibly emerged. But we know where life was found early on. And if we have to make a choice where we're going to send a rover to Mars, for instance, that's going to cost us lots of money. And so we have to think like, where do we have the highest likelihood for success? So we do have to use what we do know about life in the universe to guide searches. And as Jessica said, there's people that study agnostic biosignatures, there's people who study life as we don't know it, and there's people who think about what are other ways that life could have emerged. It's also important to say that, as Jessica said, there's different theories for how life emerged on Earth. So there's not, you know, some people might think one, some might think another. So there's a lot of research going into those different areas that we think about. But if we think about all the evidence that we have for early life on Earth and where it's found, we should use that as a guiding principle for where we're looking for life elsewhere in bodies.
Great answers. So Judith from the Netherlands is asking, do you ever have to write your own data analysis, simulation, or statistics tools and software to make up for missing conditions or unknown variables?
So I am not a computer person. I think there's a lot of really fantastic modelers. I am nowhere near one of them, but I love collaborating with people who model and do theoretical simulations. Like I said earlier, I think it's going to take all kinds to answer these questions. There's a lot of theories and a lot of components. And so, of course, and again, like I said, we're very, we can do a lot of good lab work, and that is like real data you can, you know, touch and hold. And so I think it's really worthwhile, but that means we can't run every lab experiment we think of. And so it's very important to talk to these modelers to say, hey, here's the 3 conditions I've ran, here's kind of what we're seeing. If we go way up here, you know, what could possibly happen? What, you know, what with your models, what do you think? And I think that this is especially also important for, you know, an earlier question was on kind of the conditions of these other worlds, right? We have, like Bonnie said, these spacecrafts are not cheap. And we, you know, maybe get a couple shots to send missions to these places. And so with that data, we have to do a lot of extrapolating and modeling. And there's a lot of very excellent work in that field, and it's really critical to our understanding.
Yeah, I also do not do that, but I do use a lot of data that builds off modeling, and I use a lot of modeling to inform how I might set up an experiment. There are a lot of people now that are starting to work on areas of large-scale data synthesis and artificial intelligence for biosignature research, and that are setting up like websites that put all the biosignatures in one area so that people can do large-scale data analysis of it, like you might have in paleontology, where people have the paleontology database and can assess, you know, millions of data points in one. So this is certainly a growing field, and it's one that's definitely needed, because if we think about missions to the outer edge of the solar system, it's going to take a long time to get the data back, and so there needs to be some sort of, you know, autonomous processing of the data so that we can think about those things.
Great. So Sarah from Mexico is asking, what do we gain as humanity from astrobiology research?
So I think there's a couple ways to answer this question. There is And Bonnie touched on this earlier, so I want to give credit for bringing it up. So first of all, research doesn't exist in a vacuum. A lot of the things that we're researching, say, like a lot of a huge part of what I do is amino acid synthesis. Amino acids are not just important because of the origin of life. They're absolutely critical for medicine, understanding life, having, your body needs these. So a lot of these molecules that we're looking at have significant overlap with pharmaceuticals, with natural products, which is all really important for chemistry research and medical research. And, Bonnie was talking about earlier how some of her research in the past, and some of this research could play into, say, stuff relevant to earth science and climate change, the modeling and machine learning tools people develop could be useful on a larger scale. So, this research doesn't exist alone and fits into like the whole world. And so there's a variety of ways that could occur, but also I would argue fundamentally understanding how we evolved and how life emerged is a huge scientific question and is worth answering just on its own, and for itself. We have a lot we don't understand about the solar system as well. So it's for ourselves and then also for going to these other worlds and exploring them and just understanding how we fit into the universe. And I think that is a worthwhile endeavor.
Yes, I agree completely with Jessica. I think that in the modern day, there's a lot of thought that all science must be commercialized or translatable to make money. And I don't necessarily agree with that. I think that fundamental questions about our existence are important for itself. Other ways that astrobiology helps the community and helps the planet. For instance, there's been studies from NASA where people go into prisons and they teach people about astrobiology, and the prisoners who've been part of those education systems are actually less likely to recommit offenses in the future. So that's a direct way that that's helping the population. Another thing is if you live in a country like I come from, where climate change is affecting us every single day, increased bushfires, increased droughts, increased floods, it's harsher and harsher to live in that country. A lot of the research into space has helped us. It's helped us make food that preserves for longer. It's helped us understand what the limits of life are. It helps us understand how we can live in these harsh conditions and what we can do to help our farmers be able to cultivate crops. There's other aspects of space research that have helped us develop LASIK, have helped us develop Velcro, which is incredibly helpful for people with disabilities. If we want to think about COVID, the technology behind the rapid COVID test comes from astrobiology. It comes from the DNA of microorganisms living in a hot spring pool in Yellowstone that were being studied as the limits of life. The thing about major scientific advancements is that often they come from something tangential. They don't come from someone setting out to solve that question. They're trying to solve a different question, and then that benefits everybody. And so I think the more we try to focus, no, we have to make sure that this is going to give us money. We have to make sure that this is going to benefit somebody. We're going to really limit the applications of what we can do.
So I agree with everything Bonnie said. And I think it is very worth, looking up. NASA developed a ton of stuff for its human spaceflight program. And there are like a ton of like BuzzFeedy like articles about how that transitioned into daily life. And I think that, yeah, we really just don't know. We don't know. And I think I completely, again, want to just echo, like, it doesn't need to be commercial. I think this is worth answering. But I think that just understanding a larger picture of science and life will give us, really useful things. And we just don't know what that entirely is yet.
Thank you. That was a really great session. I think since we're at the top of the hour, we will stop here. So I think that Prachi and I will stay on for a few minutes to talk through some of the bookkeeping, but I'd like to thank both Bonnie and Jessica for sharing like such amazing stories and insights. I really enjoyed it, and I know them better than anyone else in this call, and I learned a ton. So I can just imagine how much you all learned. So thank you both for sharing an hour of your time with us. We really, really appreciate it. I'm really grateful.
Yeah. Thank you, Jessica, again, and thank you, Bonnie, and thank you, Tess, for bringing them. Bonnie, I did want to iterate your last point itself about fundamental science and the applications which we may not know what it will yield at the end. So one of my favorite things in DC to do is go to the Golden Goose Awards, and I'm going to put that on Discord. If you're ever in Washington, DC, these are the awards to come to in September. These are awards given to big scientific research that was federally funded maybe 5, 10, 20 years ago, but today has yielded big technology breakthroughs. So in fact, the inventor of LASIK was given this award, but initially it was, well, someone exploring, well, what do we do with these lights, and how do our eyes work? One of my favorite examples was this one group that was given funding to massage rats. And that sounds very funny. Why are we giving funding to massage rats? Turned out they realized that when you massage baby rats, especially premature rats, so those rats who are born prematurely, that even though they're premature, they're small, much weaker than kind of normal babies. They caught up on their growth really quickly. And today we use the same technique for premature human babies, where if they are massaged and handled well, their time they spend in incubation is very, very little. They don't have to be hospitalized for long days. And their growth rates are just like normally born babies in a matter of days. So again, vague scientific research sounds very funny. Why study microorganisms at the bottom of Yellowstone Park? But again, can have huge consequences. So thanks for highlighting that really key point.
All right, thank you, both of you. So you're welcome to leave or stay on as you wish. Thank you again for joining us. And we'll now transition over to talking about some bookkeeping stuff.
Thanks for having
Yes.
us.
Thank you, Bonnie.
Okay,
All
so
right.
I just wanted to touch base on a few things. So you have until this Sunday to submit the assignment on Canvas. So hopefully everyone found it. It's under modules at the bottom, or I don't remember where it is. It's somewhere under modules though. If you have problems finding it, we can direct you on the Discord. And then it's a very simple assignment, the full names and the email addresses of your team members and where you're going, and then just a certification that someone will be there to present live at the semifinals and the finals. If you're still looking for a team, especially some of the people who just have been very recently signing up, you can continue to form teams. It's just that after Sunday the 13th, we can't take new teams. And that's just because we will need to start moving forward with organization, and we won't be able to be adding teams after the 13th. And it's also like, so we suggest 3 to 5 people on a team, but if you happen to want to have a smaller team of 1 or 2 people, like that's fine as well, but just no more than 5. Prachi, is there anything else?
No questions. How is everybody feeling about their team members? And you have all made teams. I hope by now, if you still need teams, there's so many people messaging on Discord. So message one of them, add them as a friend, make your own chat. I don't think you need to add them as a friend either. Maybe Ananya can tell you more here if she has any ideas for you to make teams. She knows how to make friends and she knows how to make teams. So otherwise, ask Ananya, how do I find a team? But I hope you guys are feeling well. For some of you, maybe late registrants, test has extended. So find a team by 13th, which is a lot of time. So again, chat with people. You could also go to introductions and see each other's backgrounds, and then maybe approach people, join the team as well if you think you really want to be in someone's team. So yeah, any questions, thoughts?
One other thing I wanted to emphasize, maybe some direction. So we are focusing on astrobiology. So this is not about bringing humans into space. As we discussed last week, that's more space biology, which is a very cool and exciting field, but it's just not the field that we are focusing on currently. So we prefer if your missions are not about bringing people to space, because that's outside of the scope of astrobiology. We also, again, want to emphasize that this is the science side of the mission. So if you're very excited and you want to like figure out all the engineering, you should go for it, and we encourage you, but we will not be judging you or really considering that during the semifinals and the finals. So we want to encourage you to be excited and do what you're passionate about, but just understand that for the judging aspect, we won't be judging that particularly. And then finally, we kind of anticipated people were spending maybe 2 hours or so a week on this portion, just as a sort of general guideline. If people were wondering like how deep and in depth they should be going. So just as a guideline. But maybe it's up to each team, of course, but try to at least reach something together so that everyone's kind of happy with what the level of investment of the team is.
Yeah, and we'll have milestones in between to help you make sure you're getting all the support you need. we'll have check-in as well. So, and next week you will see and hear from some speakers who design missions too. So you'll get some nice inspiring ideas. And I'm so curious, Serene, where you are, because you are in a lab and I feel like I should ask you for a lab tour.
I have to run now, but I will leave this open, or does anyone else have anything to say?
Yeah, do you guys have last minute questions, thoughts? Okay, everyone is just hanging out. Okay, go find a team, ping us, but otherwise you can find a team. You're all wonderful people. I'm sure everyone wants to be in your team. So make a team, submit the Canvas quiz by the 13th, and next week we'll have more fun.
All right, I'm going to end the call. So goodbye, everyone. See you next week.
Thanks all.